What up, what up, what up? Welcome back to another video of biology of cells and tissues. We are approaching the end. We've got two more to get through. That is going to be the reproductive systems. We're going to start with the female reproductive system, and then afterwards we'll look at the male reproductive system. The female is a little bit more, I don't want to say complex compared to the male. Let's just say that the male is less detailed. We'll go with that. So the female reproductive system, we're going to look at a whole bunch of different components. We're going to be looking at the different components of the reproductive system. Of course, we're going to look at the ovaries, the fallopian tubes, corpus luteum, uterus, all sorts of good stuff. I have also, from the original structure of this PowerPoint of the professor who uses it, I branched off of it a little bit more and went into a little bit deeper detail. The reason being is that in the future, specifically in a little bit in physiology, and then again, so physiology 2 in trimester 3, and then again in physical diagnosis in tri 4, and then again in your OBGYN and pediatrics course, you're going to talk, I believe that is tri 6. It's either tri 5 or tri 6. You're going to dive into the female reproductive system, and I felt generally underprepared for this topic in those future courses. So I wanted to make sure I give you all the information here so that way you can come back, look back at this, and you have it all, you're exposed to all of it now for the future. So in general, the reproductive system, it's to produce female gametes. Gametes, when we talk about embryology, we know that a gamete is going to be the half of our DNA that, when put together with the sperm, is going to create the zygote or the conceptus. Conceptus, that's another word for zygote. It's just when the male and the female gametes meet together to form a full, a cell with a full set of chromosomes. Okay, so the components, we have the ovaries, the oviducts, aka uterine tubes, aka fallopian tubes. Any of those words you're going to see, it's just fine. And the uterus, and then the vagina. So here is our reproductive organ overall. We have the ovaries here. We have the fallopian tube, the uterus, and then the vagina with the cervix separating the vagina from the uterus. As well, there are a lot of ligamentous type structures. This is one of the areas where, if you want to be a chiropractor who focuses and works a lot with the prenatal or the perinatal women or pregnant women in general, this is going to be a lot of stuff. You really want to know this very well, because there's a couple of different techniques, and I recommend that you shadow some different mom type doctors, not like doctors who are mothers, but chiropractors who work with a lot of mothers, pregnant mothers especially, because there are some really cool techniques that you can use for, like ligament releasing ligaments such as the broad ligament, as well as there are some suspensory ligaments as well that tend to get very tight and can cause a lot of pain and discomfort during pregnancy. So keep that in the back of your mind, and look for those opportunities to meet those kind of providers, as well as shadow them. Okay, so we're going to start with the ovaries. So in general, the outermost layer, they have a germinal epithelium. This is a simple cuboidal epithelium. It's just a single layer of cells all the way around the entire ovary. We also have the next layer, we have a tunica albuginea. This is a little bit deeper. It's a thin layer. It is poorly defined, dense connective tissue for our structural support. So we can see that germinal layer on top. tunica underneath. Much like our kidneys, we divide the ovary into two layers. We divide it into a cortex, which is the outer chunk layer, and then the medulla, which is the inner chunk layer. The cortex is the outer region. It contains connective tissue as well as ovarian follicles. We're going to get all into ovarian follicles, what they do, their different stages, in a little bit. The medulla is the inner region, and it has loose connective tissue, blood vessels, lymphatics, nerves, all of that good stuff. It is the vascular and neural support to the ovarian structures. Talking about follicles, the follicles, these are what become the egg of that is released during ovulation. Okay, you'll also hear the word oocyte. That is also referring to that specific egg that's going to be released during ovulation. So an ovarian follicle, this is a small fluid-filled sac in the ovary that contains an immature egg. Follicles are a vital part of women's fertility and are involved in the menstrual cycle, ovulation, and early pregnancy. Here are all the different types, and these are in order of what they're going to become. Primordial, primary unilaminar, primary multilaminar, antral or secondary follicle, a Graafian or tertiary follicle, and then an atretic follicle. We'll cover each of those. You can come back to this a couple times if you want, but I've also taken each of these images and inserted them into the appropriate slides. So, primordial follicles. This is the earliest stage of follicle development, and it's important to understand women are born with all of the primordial follicles, so all of the potential eggs that they can have. The women are born with them right away. So they always have all of the potential eggs that they could possibly have. And they are in this state of early development, and they contain what's called a primary oocyte that is actually going to be paused in its division process. So basically, when a female is being formed inside of the womb and the ovaries are formed, all of these primordial follicles form, and then they all hit the pause button. So that way you can have, I believe the numbers where you can have, I think there was 200 to 300,000 primordial follicles. I believe that was the number that I most recently read about. So you can have a lot of these follicles, but they're all paused. Some of the defining characteristics: this primary oocyte, that was going to become the egg, is surrounded by a single layer of flattened follicular cells. And these follicles are going to remain dormant until a hormone called follicle-stimulating hormone starts to be released at puberty from the anterior pituitary gland. And we'll look at all of the different hormones and what releases what and how it interacts with everything. So here we have these follicular cells. So we can see kind of here, this is what we're going to talk about next, these primary follicles. But you can see these follicular cells here, how flat they are. They're all squished down. These are our primordial follicles compared to our next step where they look much more cuboidal. So our primary unilaminar follicle, laminar, this specifically means like layers. So laminar, so unilaminar, single layer. This is the primary oocyte, that little egg portion, is surrounded by a single layer of cuboidal follicular cells. And this is going to mark the transition from the primordial follicle to the early primary follicle stage. Here we have it all different labeled. We have the nucleolus, the nucleus. This whole structure is the oocyte. We have that simple cuboidal epithelium surrounding it as well. Next step, the primary multilaminar follicle. So just as we had unilaminar, now think multilaminar follicle. We are going to have multiple layers of the follicular cells. Sorry, let me move my camera just a little bit so we can see the full screen. There we go. So that primary multilaminar follicle. So the primary oocyte has 2 or more layers of cuboidal follicular cells. The innermost layers, so that very first layer that are in contact with the oocyte, they form these gap junctions, and we call that layer granulosa cells. These cells are going to come back a lot, so make sure that we stick with these. And as well, throughout this presentation from here on out, if you see the word follicular cell, you can substitute it with granulosa cell. Follicular cell, granulosa cell, granulosa cell, follicular cell. They essentially mean the exact same thing, so you can kind of connect those two things together. There's also a region called the zona pellucida. This is a glycoprotein-rich extracellular matrix that surrounds the oocyte. And it is required, it's produced by the oocyte as well as those granulosa cells, and it's required for sperm binding and initiating the fertilization process. Then we also have a structure called the follicular theca. This is composed of a modified ovarian stroma. Remember when we talk about stroma versus parenchyma? The stroma is going to be more of the connective type tissues, not the functional tissues. So we have the theca interna, which is made primarily of vascular connective tissues, and then the theca externa, which is composed of fibrous connective tissue. The theca interna is going to come back as well. We're going to talk about it a little bit, how it produces hormones called androgens, and androgens are precursors for estrogen. We mentioned androgens briefly when we talked about the suprarenal gland or the adrenal gland, how one of the layers of the adrenal gland produces the androgens, or produces the DHEA, which can then also be converted into estrogen. So the theca interna also plays a role in estrogen production. Here we have all these different structures. The zona pellucida is immediately outside of that oocyte. Then we have our multilaminar follicle all around all these different layers. We also have basement membrane. We call this the granulosa layer. Here's just a couple of more pictures of this you can kind of get into it. So if we're pointing like this pink layer right in here, that's going to be our zona pellucida. This whole circular structure is the oocyte. More defined, that is the nucleus in there, and then the granulosa layer out here. I thought that I had a picture with the theca interna and externa. I probably do in the lab material, so if you want to go and check those out, see if there is an image with those labeled on it. Quick break for clinical application. So the growing primary follicles, so the one follicles in this stage, can become involved in a condition called polycystic ovary syndrome, or PCOS. This has enlarged ovaries, and you get these ovary cysts, as well as a non-ovulatory state. So essentially the follicles grow, they start to grow, they get to this point, but they never actually reach full maturity. So you can have a lot of issues with infertility with women with PCOS. This is a continuously rising concern in the United States right now. A ton of women are experiencing PCOS. There are some articles and some studies out there that suggest there is a linkage to modern birth control, specifically like the hormonal pills that a lot of people have been taking. So I suggest that you, as you go through the program, dive into that a little bit, because you will see a lot of patients, especially if you're working with women who have PCOS or PCOS-like symptoms or conditions. So it's a very good condition to dive into and to understand the complexities of this. Okay. Continuing with the follicles, the next step is an antral, or a secondary follicle. Let me just double check for some reason. Yeah, we wrote it in there. Good. Wanted to make sure everything looked nice. So the antral, or the secondary follicle, it's called secondary because now it's a secondary oocyte. We go from a primary oocyte to a secondary oocyte. So specifically, the oocyte inside of the whole structure is developing. So you get this fluid that's called liquor folliculi, starts to accumulate between the granulosa cells. It's actually secreted by the follicular cells. Again, we can substitute follicular for granulosa at any point in time, so by the follicular or the granulosa cells. And it contains nutrients, hormones, as well as growth factors. This fluid leads to the formation of a fluid-filled cavity called the antrum. You can see this antrum starting to form here. You also get a structure called cumulus oophorus. This is a cluster, also called a hillock or hillock. Cluster means hillock, of follicular, the granulosa cells that actually anchor the oocyte to the wall of the follicle. Don't believe it is labeled on this. I thought I had. Here we go, CO. Yeah, there it is right there. The CO layer right here. This region over here is that cumulus oophorus. Another picture, here's all of our different layers. Here we have these labeled: the theca interna, the theca externa, the stroma, basement membrane layer, granulosa, and then the antrum. So these are good pictures to kind of practice on. A lot of our lab stuff is just raw memorization, so you really just need to click through enough of these and label enough of these to be able to tell the difference between them. These, I personally feel like, don't have as distinct, easily obvious, or as obviously differentiable layers just by looking at them. So continuing. Here is our secondary follicle. There's the antrum. They have the zona granulosa, the zona pellucida still immediately around the oocyte, all that good stuff. Another picture. Spend some time looking at those. So our follicle continues to develop, and finally it becomes a Graafian, or a tertiary follicle. This is the most mature follicle. The antrum is fully formed, occupying a large portion. The oocyte completes the first meiotic division and becomes a secondary oocyte. I do apologize, I misspoke, jumping back really quickly. This is the secondary follicle. We had a primary follicle. This is now the secondary follicle. Not until the Graafian follicle do we have a secondary oocyte. My apologies. So this is a tertiary follicle with a secondary oocyte. Okay. We also see another structure called the corona radiata, and these are the cells that actually remain in direct contact with the oocyte across the zona pellucida, and they're actually going to be released with the oocyte during the process of ovulation. So just a couple more pictures. Kind of, there's a corona radiata. You can see the darker layer of cells right around the ovum, or the oocyte, right there. There's our antrum, all that good stuff. Here's another picture of all of that. So all of this whole process is meant to create a follicle with which that we are going to be able to release. It's going to be released during ovulation to potentially be fertilized by a sperm and then develop into a fetus. During this whole process, our body starts this process with multiple follicles. So you can have multiple primordial follicles begin this process of turning into primary, secondary, tertiary type follicles. However, one is going to become the dominant one. And that dominant one is going to be the one that goes fully on to become fully matured and then eventually be released into the fallopian tube. This is one of the ways that you can have fraternal twins, is if this process, if you potentially get multiple tertiary follicles released, then you can have the possibility of twins. However, in most cases, all of the follicles that are not determined to be the primary ones go through a process called atresia or degeneration. So you get an atretic follicle. These are the follicles that degenerate and die without completing development. This can occur at any stage, all the way from the primordial follicle up to an antral follicle stage. So once it reaches the tertiary stage, that Graafian follicle, it no longer is this is not going to happen. It's not going to just degenerate and die. But any time before that, it can enter into this process and regress. Just a picture of that whole process going through from, I'm actually going to move this one up. So you can see it going through all the different types of follicles as they mature. Here's just the summary of development, kind of how, I'm not going to read through all of this because we literally just went through all of it. But this is kind of the condensed, easy, quick picture of the development of each of these components and how they add on to the next. The next step, ovulation. So during ovulation, that Graafian follicle, that tertiary follicle, is actually going to rupture and release the mature egg from the ovary, and everything that's left behind transforms into a structure called the corpus luteum. So this whole structure, the whole follicle is going to burst, release the egg, and then it turns into the corpus luteum, which is actually why we'll see within the menstrual cycle we have a follicular phase and then a luteal phase. The follicular phase is when the follicle is all growing, and then once ovulation happens, we enter a phase called the luteal phase. The corpus luteum is made up of the follicular and the thecal cells, and it remains in the ovary undergoing a process called luteinization. This is stimulated by another hormone released by the pituitary gland called luteinizing hormone, or LH is how we shorten it. And as a result, the corpus luteum becomes a temporary endocrine gland secreting progesterone and estrogen. The granulosa lutein, that's derived from the follicular cells, makes progesterone, and then the theca lutein from the theca interna produces estrogen. And without pregnancy, this phase, the corpus luteum, will be active for about 10 days and then regresses to form a structure called the corpus albicans. The corpus albicans is essentially a dying corpus luteum, or a degenerating corpus luteum. However, if we do have pregnancy, so the oocyte, that ovum, gets fertilized and starts to get implanted, the corpus luteum will actually remain active for about 5 months, and produce and it will be maintained by the hormone HCG, human chorionic gonadotropin, that is secreted by the placenta that's created. And the progesterone maintains the uterine level, and then we'll also start to release a hormone called relaxin. This is to prepare the body for childbirth. The closer we get to the end stages of pregnancy, the more relaxin that our body produces. And it's actually designed to, it softens the pubic symphysis as well as the other pelvic ligaments to allow the pelvis to expand and open to allow for childbirth. So here's that corpus luteum. We have the granulosa cell, and then the theca luteal cells. The corpus albicans, as I said, this is the degenerating corpus luteum, and these actually remain behind. So it looks like a little fibrous, whitish scar tissue within the ovary. And these, you can actually see these, you can see these with a microscope. So after a person, as I say, after a person passes, if you're doing if you're dissecting this individual and you take a slice of the ovary, you can actually determine the history of ovulation and reproductive health of this individual by looking at the corpus albicans, like the quantity of them, the location of them, and the kind of their general structure. So it's kind of a cool little thing that you can look at. There's our corpus albicans. You can see it's literally just kind of a ball. There's no real cellular structure to it because this is not functional. No, it doesn't do anything. It's basically like a little tiny nugget of scar tissue. Okay, we're approaching the 27-minute mark. What I'm going to do is I'm going to go ahead and pause at this point, and we're going to call this part 1. And then we'll continue in part 2 talking about the uterine tubes, aka the fallopian tubes. As always, thank you so much for watching. Have a good day and good luck studying.